Hi, my name is Charlotte Aumeier and I'm a biochemistry professor here at the University of Geneva. My background is not pure biochemistry. I actually studied biology with a big focus on botany. I studied gravity sensing in plants and then did my PhD on the cytoskeleton of diatoms. And the cytoskeleton is really what fascinated me early on, already during my bachelor, and is still fascinating me. So what is really the source of the fascination? In the beginning, it was the pure beauty of the cytoskeleton. If you look in a microscope and you observe the cytoskeleton, it's just so organized and there are so many different cytoskeletal components, and it makes a very dense network in the full cell. I was always puzzled, and I'm still puzzled, how all these different components can orchestrate in space and time to fulfill many different biological functions. The cytoskeleton is not only present in all cells, it's also involved in so many different processes. They are involved in cell division, cell migration, shaping the cell, force generation, and also organizing the cell from within. The cytoskeleton, one component which we study the most, the microtubule cytoskeleton, can serve as a street network to transport cargo within the cell. In the cell, you have motor proteins, which are like cars, which use the microtubule cytoskeleton to transport the cargo in a directed manner. My group started this transport of a motor protein along this microtubule, and we discovered that this motion of motor can actually regulate the underlying street network. So the car can control which street it needs most to be stable in space and time. Based on this finding, we now got a generous funding from the SNS starting grant, which is currently a replacement for the ERC grant here in Switzerland. What we aim to study further in this project is to, first of all, develop chemical probes, which allows us to visualize certain processes in the cell. We want to understand in the cell, but also in vitro, how the interaction of motor proteins and the microtubule really change the dynamic properties, and then if this can further lead to a polarization of the network and then to reshaping of the cell. This research is embedded here very well at the University of Geneva because we have a lot of interaction with other professors and groups studying the cytoskeleton. In my department, we have a great interaction from the biochemistry sides, and we can also bridge to the physics department to understand the physical principle of the biological properties we observe. So if you're eager to join my great team of PhDs and postdocs, which really drive this project, please contact me because we are continuously looking for new students.